Welcome to our exploration of gene theory, the scientific concept that revolutionized our understanding of life. Gene theory proposes that genes, the fundamental units of heredity, are responsible for passing traits from one generation to the next. This idea has shaped modern biology, medicine, and even agriculture. In this video, we'll journey through the origins, development, and impact of gene theory, discovering how it has unlocked the secrets of life itself. Let's begin by understanding what genes are and why they matter so much in the grand tapestry of living organisms. Genes are segments of DNA that carry the instructions for building and maintaining an organism. Think of them as the blueprints for life, dictating everything from eye color to susceptibility to certain diseases. Each gene contains specific information that determines how our bodies grow, develop, and function. These tiny sequences are found within the chromosomes in the nucleus of every cell. Understanding genes is crucial because they are the foundation upon which all living things are built, influencing both our similarities and our unique differences. Journey to understanding genes began in the 19th century with Gregor Mendel, an Austrian monk. Through his experiments with pea plants, Mendel observed how traits were inherited in predictable patterns. His work laid the groundwork for gene theory, even though the term gene hadn't been coined yet. Mendel's meticulous crossbreeding experiments revealed that traits are passed down in discrete units, which we now know as genes. His findings were initially overlooked but later recognized as foundational to genetics. Mendel's experiments involved cross-pollinating pea plants with different traits, such as flower color and seed shape. He noticed that certain traits would appear in predictable ratios in the offspring. This led him to propose the existence of factors, what we now call genes, that control inheritance. Mendel's laws of segregation and independent assortment became the cornerstones of classical genetics, demonstrating that inheritance is not a blend, but rather a transmission of distinct units. As science progressed, researchers discovered that genes are located on chromosomes, structures within the cell nucleus. The chromosome theory of inheritance, developed in the early 20th century, connected Mendel's abstract factors to physical entities. This breakthrough helped scientists visualize how genes are organized and passed on during cell division. The realization that chromosomes carry genes provided a physical basis for heredity and further solidified gene theory's place in biology. Next major leap came with the discovery that DNA is the molecule that makes up genes. In the mid-20th century, experiments by scientists like Avery, Hershey, and Chase demonstrated that DNA, not protein, is the genetic material. This finding was pivotal as it explained how genetic information is stored and transmitted. DNA's unique structure allows it to replicate and carry instructions for building proteins, the workhorses of the cell. 1953, James Watson and Francis Crick unveiled the double helix structure of DNA, a discovery that transformed biology. Their model showed how DNA's two strands are held together by complementary base pairs, enabling accurate replication. This elegant structure explained how genetic information could be copied and passed on with remarkable fidelity. The double helix became an iconic symbol of life's complexity and the power of scientific discovery. Genes function by providing the instructions for making proteins, which perform most of the tasks in our bodies. Each gene codes for a specific protein, and these proteins determine everything from our physical traits to how our cells operate. The process involves two main steps: transcription, where DNA is copied into RNA, and translation, where RNA is used to build proteins. This flow of information is known as the central dogma of molecular biology. Central dogma of molecular biology describes the flow of genetic information. DNA makes RNA, and RNA makes protein. This concept is fundamental to understanding how genes control the functions of living organisms. It explains how genetic instructions are converted into the molecules that build and maintain our bodies. The central dogma highlights the precision and complexity of life at the molecular level. Genes are not static. They can change through mutations, which are alterations in the DNA sequence. Some mutations have little effect, while others can lead to significant changes in traits or even cause diseases. Mutations are also a source of genetic variation, providing the raw material for evolution. Without mutations, populations would lack the diversity needed to adapt to changing environments. Genes are inherited in specific patterns, which can be observed in families and populations. Some traits are controlled by a single gene, while others involve multiple genes working together. Mendelian inheritance describes simple patterns, such as dominant and recessive traits, while more complex inheritance involves interactions between genes and the environment. Understanding these patterns helps us predict the likelihood of certain traits appearing in offspring. Sometimes, changes in genes can lead to genetic disorders. These conditions are caused by mutations that disrupt normal gene function. Examples include cystic fibrosis, sickle cell anemia, and Huntington's disease. Studying genetic disorders has helped scientists understand the importance of genes in health and disease and has paved the way for new treatments and therapies. Genes play a central role in evolution, the process by which species change over time. Natural selection acts on genetic variation, favoring traits that enhance survival and reproduction. Over generations, beneficial mutations accumulate, leading to the adaptation of populations. Gene theory provides the molecular basis for evolution, connecting the inheritance of traits to the diversity of life on Earth. Today, genetics is a rapidly advancing field thanks to new technologies and discoveries. Scientists can now sequence entire genomes, edit genes with precision, and study how genes interact with each other and the environment. Modern genetics has applications in medicine, agriculture, and even forensic science, demonstrating the far-reaching impact of gene theory. One of the most ambitious scientific projects ever undertaken was the Human Genome Project. Launched in 1990, its goal was to map all the genes in human DNA. Completed in 2003, the project provided a comprehensive blueprint of our genetic makeup. This achievement has transformed medicine, enabling personalized treatments and a deeper understanding of human biology. Gene editing technologies like CRISPR have revolutionized genetics. CRISPR allows scientists to make precise changes to DNA, opening up possibilities for treating genetic diseases, improving crops, and even eradicating certain inherited conditions. While these advances offer tremendous promise, they also raise important ethical questions about how we use and control genetic information. Personalized medicine uses genetic information to tailor treatments to individual patients. By understanding a person's genetic makeup, doctors can predict how they will respond to certain drugs, identify risks for specific diseases, and develop targeted therapies. This approach promises more effective and safer treatments, marking a new era in healthcare. Gene theory has also transformed agriculture. Scientists use genetic knowledge to develop crops that are more resistant to pests, diseases, and environmental stresses. Genetic engineering allows for the creation of plants with improved nutritional value, higher yields, and better adaptability. These advances help ensure food security for a growing global population. Genetic analysis has become a powerful tool in forensic science. DNA profiling can identify individuals with incredible accuracy, helping solve crimes and exonerate the innocent. The ability to match genetic material from crime scenes to suspects has revolutionized law enforcement and the justice system, making gene theory an essential part of modern investigations. Many people are now using genetic testing to learn about their ancestry and family history. By analyzing specific markers in DNA, scientists can trace lineage and migration patterns over thousands of years. This has opened up new ways for individuals to connect with their heritage and understand the journeys of their ancestors. But genetics is the study of changes in gene expression that do not involve alterations to the DNA sequence itself. Environmental factors such as diet and stress can influence how genes are turned on or off. Epigenetics adds another layer of complexity to gene theory, showing that our experiences can affect how our genes function and even be passed on to future generations. Not all genes are active at all times. Gene regulation is the process by which cells control which genes are turned on or off, depending on the needs of the organism. This regulation ensures that the right proteins are made at the right time and in the right amounts. Understanding gene regulation is key to unraveling the complexities of development, health, and disease. Cancer is often caused by mutations in genes that control cell growth and division. When these genes malfunction, cells can grow uncontrollably, forming tumors. Research into the genetic basis of cancer has led to new treatments that target specific mutations, offering hope for more effective therapies and better outcomes for patients. Genes guide the development of organisms from a single cell to a complex being. They control processes like cell division, differentiation, and organ formation. By studying how genes orchestrate development, scientists gain insights into birth defects, growth disorders, and the incredible diversity of life forms. Research suggests that genes can influence behavior, personality, and even intelligence. While environment and upbringing play significant roles, certain genetic variations are linked to traits like temperament and risk for mental health conditions. The interplay between genes and environment shapes who we are, highlighting the complexity of human behavior. Intelligence is a complex trait influenced by many genes and environmental factors. Studies have identified certain genetic variants associated with cognitive abilities, but no single gene determines intelligence. The relationship between genes and intelligence is intricate, involving interactions among multiple genes and life experiences. Some people are naturally resistant to certain diseases due to their genetic makeup. For example, specific gene variants can provide protection against infections like HIV or malaria. Understanding these genetic factors helps scientists develop new treatments and preventive strategies, improving public health worldwide. Aging is influenced by both genetic and environmental factors. Certain genes are associated with longevity and the aging process. By studying these genes, scientists hope to uncover ways to promote healthy aging and extend lifespan. The quest to understand the genetics of aging continues to inspire research and innovation. Genes interact with the environment in complex ways. While genes provide the blueprint, environmental factors like nutrition, exposure to toxins, and lifestyle choices can influence how genes are expressed. This dynamic relationship shapes our health, development, and risk for various diseases. Power to manipulate genes raises important ethical questions. Issues such as genetic privacy, designer bodies, and the potential for discrimination must be carefully considered. As genetic technologies advance, society must balance the benefits with the risks, ensuring that progress is guided by ethical principles. Genetic counseling helps individuals and families understand their genetic risks for certain conditions. Counselors provide information, support, and guidance on genetic testing and its implications. This service is especially important for people with a family history of genetic disorders or those considering starting a family. Gene therapy is an innovative approach to treating genetic disorders by introducing, removing, or altering genes within a person's cells. This technique holds promise for curing diseases that were once thought to be untreatable. While still in its early stages, gene therapy represents a major leap forward in medicine. Genetic engineering is not limited to plants, it's also used in animals. Scientists can modify animal genes to study diseases, improve livestock, or even produce medicines. These advances have significant implications for agriculture, medicine, and our understanding of biology. Synthetic biology combines engineering and biology to design and build new genetic systems. Scientists can create artificial genes and organisms with novel functions, opening up possibilities for new medicines, materials, and energy sources. This cutting-edge field pushes the boundaries of what life can be. Biotechnology harnesses the power of genes to solve real-world problems. From producing insulin for diabetes to creating biofuels, genetic technologies are transforming industries. The integration of gene theory into biotechnology has led to innovations that improve health, sustainability, and quality of life. Genetic research is vital for conservation efforts. By studying the genetics of endangered species, scientists can develop strategies to preserve biodiversity and prevent extinction. Genetic tools help track populations, manage breeding programs, and restore habitats, ensuring the survival of threatened species. Microorganisms like bacteria and viruses have their own genes, which can evolve rapidly. Studying microbial genetics helps us understand disease outbreaks, antibiotic resistance, and the development of new vaccines. Microbial genetics is a key area of research with direct implications for public health. Scientists have begun to create synthetic life forms by assembling genes from scratch. These synthetic organisms can be designed for specific purposes, such as cleaning up pollution or producing valuable chemicals. The creation of synthetic life challenges our definitions of life and raises new ethical questions. Artificial intelligence is being used to analyze vast amounts of genetic data, uncovering patterns and relationships that would be impossible to detect manually. AI accelerates genetic research, aids in disease diagnosis, and helps predict the effects of genetic changes. The synergy between AI and genetics is opening up new frontiers in science. The future of medicine will be shaped by our understanding of genes. Advances in genomics, gene editing, and personalized therapies promise to revolutionize healthcare. As we continue to unlock the secrets of our DNA, we move closer to a world where diseases can be prevented, treated, or even cured at the genetic level. Genes play a significant role in shaping who we are, but they do not define us completely. Our identities are the result of a complex interplay between genetics, environment, culture, and personal choices. Understanding gene theory helps us appreciate both our shared humanity and our individual uniqueness. The impact of gene theory extends beyond science and medicine. It influences how we think about health, family, and even social issues. As genetic knowledge becomes more accessible, it's important for society to engage in thoughtful discussions about its implications, ensuring that advances benefit everyone. While gene theory has transformed biology, it's important to recognize its limits. Not all traits are determined solely by genes, many result from complex interactions with the environment. Ongoing research continues to reveal new layers of complexity, reminding us that life cannot be reduced to genes alone. Gene theory will continue to evolve as new discoveries are made. Emerging fields like epigenetics, systems biology, and synthetic biology are expanding our understanding of how genes work. The future holds exciting possibilities, from curing diseases to creating new forms of life, all built on the foundation of gene theory. Gene theory has fundamentally changed our view of life, providing a framework for understanding heredity, evolution, and disease. Its legacy can be seen in countless scientific advances and the promise of a healthier, more informed future. As we continue to explore the mysteries of genes, we celebrate the curiosity and ingenuity that drive scientific discovery.